All right. Looks like Dr. Corley is not living the naked mole rat life in a burrow. He's out on the beach. So this is Dr. Michael Corley. It's my pleasure to introduce you. Dr. Corley is an associate professor in medicine within the Division of Geriatrics, Gerontology, and Palliative Care at the Stein Institute for Research on Aging. He received his undergraduate degree from UCSD and an MA and PhD in behavioral neuroscience from the University of Hawaii and completed postdoctoral training at the John A. Burns School of Medicine in epigenetics, immunology, and infectious diseases. Dr. Corley leads a global epigenetics research program focusing on understanding the interplay between the immune system, viruses, and aging. His laboratory is at the forefront of generating critical data to advance the development, validation, and implementation of novel biomarkers and therapeutics aimed at promoting longevity. So, welcome, Dr. Corley.
Thank you, Dr. Benimo. And today, I wanted to share a little bit about the science of aging, and tell you about the field of epigenetics and other hallmarks of aging. And coming from a viewpoint of the aging community at large, focusing on the science and drivers, potentially, of mechanisms that groups are thinking about targeting for aging processes and how that might apply to people living with HIV. These are my financial relationships. So for today's learning objectives, hopefully after today, you'll be able to come away from the presentations with a summary of the geroscience hypothesis and other hallmarks of aging. We'll hopefully be able to describe the development of epigenetic clocks, which have been receiving a lot of attention in the popular press in regards to a way potentially to tell us how well we're aging by measuring the biology of aging with some molecular assays that are run in the laboratory. And then briefly, I'll highlight the current utilization of these epigenetic clocks in HIV care and people living with HIV, and tell you a little bit about some of the ongoing work that's going on in terms of potentially thinking about how these potential biomarkers may be used in the clinic or potentially need further development before they're applied out in the community with people living with HIV in terms of clinical care. So what is geroscience? The geroscience field itself is really the study of genetic, molecular, and cellular mechanisms that make aging a major risk factor and driver of common chronic conditions and diseases of older people. And really, the field of geroscience is built on the idea that rather than studying diseases as siloed mechanisms, the aging process itself is a major driver of myriad of chronic diseases. And the thought process is that if we could start to understand aging biology, this will provide insight into all diseases that are chronic conditions that may have an underlying driver that is a mechanism related to the aging process. In terms of the geroscience hypothesis, it essentially states that slowing the aging process can improve health and delay the onset of chronic diseases. When we start to think about this in terms of an individual's chronological lifespan, we start to see essentially that environmental exposures, poor diet, social stressors, economic stress, and infections such as HIV accelerate or drive this deviation from chronological age in terms of biological age. And the thought process is that the faster or the more deviance you see, or deviation you see from your chronological age, essentially this will lead you to a higher incidence of comorbidities. And in people with HIV, this has been observed in terms of very well-known epidemiological studies. When we start to look at the geroscience hypothesis, this really gets into the concept of prevention and things that people can do, such as exercise and diet, essentially at an earlier age to prevent essentially this increase in the incidence of comorbidities over time. And the geroscience hypothesis is really built on targeting aging at a particular time in life when we think that potentially this would be the moment that essentially we could slow down this process. The geroscience hypothesis essentially is not aligned completely yet with the idea that essentially we can achieve immortality, but essentially that we're on this continuous trajectory of aging and we can make modifications that essentially can accelerate the movement across time, or we can decelerate this process of aging. And the hope is that the geroscience field can identify potential agents or interventions that can slow the aging process and essentially improve health and improve the number of years people spend without disease. So essentially, an increase in health span is really the focus of the geroscience hypothesis. In terms of how we think we're going to do this, the aging field is really focused on identifying biological hallmarks of aging. Here's a summary plot from a recent review from the geroscience field, identifying these hallmarks of biological aging that are mechanisms in the lab that groups have been studying. And we have seen a lot of these in the HIV field studied in terms of impacts of the virus itself, or essentially other features of inflammation driving essentially perturbations or disturbances in these hallmarks of aging. And the aging field has really been focused on these hallmarks of aging in terms of trying to understand how we can improve health span. In terms of our research, we've really focused on epigenetic alterations, and I'll tell you today particularly about how these have led to the development of biomarkers for aging, which have been referred to as epigenetic clocks. There's been a lot of work on genomic instability. So essentially the ability of our body to maintain our DNA sequence in a particular state that occurred at a younger age, and how this potential process of stability is lost over time is a focus in terms of biological hallmarks of aging. Telomere attrition, which is essentially the shortening of telomeres. And in regards to the HIV research field, this has been a large focus as well in terms of looking at the impact of HIV in terms of telomeres and length. The loss of proteostasis, so essentially our ability to regulate the protein dynamics within our bodies. Disabled macroautophagy, so the clearance essentially of cells that are dying within the body. Deregulated nutrient sensing, which is essentially our ability to regulate our metabolic environment and understand potentially how cells are able to sense and regulate their metabolism. Mitochondrial dysfunction, which is the major producer of energy in our body in terms of these mitochondria and how potentially they can be perturbed in the aging process. This has been also a large focus in terms of hallmarks of aging. Cellular senescence, which is essentially the immune system's ability to continuously produce adequate responses becomes perturbed. And essentially, these cells are remaining in the body, but are unable essentially to have their full capacity to produce responses. Stem cell exhaustion. So cells within the body that would be able to generate and repopulate cells are essentially lost this pull. So essentially, we lose some plasticity and ability to regenerate particular compartments within the body. Altered intercellular communication, so certain parts of the body not being able to communicate with other parts of the body. The one that has been probably the most studied had been the gut-brain axis, so essentially our GI tract not being able to communicate adequately, potentially with the central nervous system. And then some of the ones that have been very well studied and very well known, such as chronic inflammation and inflammaging and dysbiosis, so essentially the perturbation of the microbiome and the loss potentially of the interplay between constituents within our gut and our overall immune system and health. And essentially the hallmarks of aging have really been a large focus for the aging field. Today, I'll tell you particularly about epigenetic alterations. So epigenetic modifications are essentially these chemical modifications that decorate our genome. Here I'm showing a graph of what a chromosome looks like, essentially where all the genes would sit that are regulating the proteins and the activity of our cells. When we see essentially what epigenetics are these chemical modifications on top of the DNA sequence itself. And essentially in every cell, these modifications are thought of as potentially these on-off switches that are decorating the genome and letting us regulate gene activity. In the lab, we have essentially the ability to measure these and to see potentially how these relate to aging and also disease states and other functional activities of the cells. In terms of the genome itself, you can start to think of these as being able to orchestrate all the potential programs or alterations that may occur as a cell is in the body throughout life. And when we start to think of this in regards to the immune system, in terms of T cell exhaustion, immune senescence, or some of the other features that have been well observed in people living with HIV, these are the marks that essentially are altered and are setting in a state that is permanent for that cell to not be able to function correctly. And these have been essentially the marks that the aging field has been using recently to be able to identify potentially which of these on and off switches are the drivers potentially of some of these aging processes that we're interested in studying and overcoming potentially with therapeutics. And here's a summary as cells age, what this would look like potentially over time. So here I'm just showing an example essentially of 20 potential genes, and we can think of these genes as genes involved in inflammation or genes involved in immune senescence. Each row is essentially where we would be looking at these on-off switches or chemical modifications on these genes. And essentially, at a younger age, we can potentially see here that these cells would be on an on state, so referred to as unmethylated in terms of this particular DNA methylation modification. And as you can see over time, essentially these light switches start to be turned off. And we start to see essentially that things become methylated. So you can start to imagine throughout time and throughout the whole genome, these decorations or light switches throughout our whole genome really become quite orchestrated in terms of the aging process. But if we essentially are flipping on and off these light switches at a quicker rate, cells start to alter a lot quicker than potentially would have occurred in a potential natural state. So you can start to see how the environment, infections, and other aspects of our life potentially could alter gene activity and set up a state essentially where we were not in a state where that cell was at a younger age in an individual's lifespan. In terms of how these have been used from the aging field, in 2013, a very landmark paper was published where it was demonstrated that essentially from two groups, the most well-known has been a group out of the University of California, Los Angeles, Steve Horvath, where they utilized data from these chemical modifications or DNA methylation from over 10,000 people, and they had access to individuals' chronological age. And what they were able to demonstrate was that they could predict how old somebody was with very high accuracy. And they referred to these as epigenetic clocks. So the epigenetic component being the measurement of these chemical modifications, and the clock component being it can tell you how old that DNA sample was biologically of that individual. At the same time, around the same time, here at the University of California, San Diego, a paper from Trey Ideker's group as well demonstrated the same thing, that they could use these methylation profiles to measure aging. And in 2013, this was really kind of the first big evidence that potentially these marks were important potentially for measuring some of the aging processes. In terms of a summary of where this currently sits, from the most well-known kind of epigenetic clocks that are these predictors of biological age, there's essentially been four that have been the most well-studied and most widely utilized. Today, I'll tell you about how these were kind of developed and what they mean. Here in this in this summary table, I'm presenting the number of these on-off switches or marks that are measured. So you can see here that 353 positions or potential marks in an individual's DNA are given very high predictive accuracy in being able to predict chronological age. And this works across tissues and cell types. You can do this with another subset of data that had been available where they were able to train and look at the lifespan of individuals. So they had a DNA sample in some of the NIH-funded studies where they were able to look at mortality outcomes. And in this particular clock, they were able to measure 1,030 marks that were quite predictive in terms of mortality risk prediction. And in this particular clock, this Grim Age clock, they were also able to include some other hallmarks of aging, such as inflammation, when they built the clock. And I'll tell you a little bit more about how this has been developed. So in this summary slide here, this would essentially be what we're looking at in terms of when someone talks about building an epigenetic clock and what they're looking at. And really, this is relied on the field of machine learning and data science that, essentially sat in the realm of biostatistics in 2013 and has now been included, I think, in the daily conversation in terms of AI and machine learning. But really what it boils down to is having access to epigenetic data with a lot of features. So essentially, as I told you, the human genome has over 23 million potential positions where you could measure these light switches or DNA methylation modifications. And this data set is trained using a machine learning model, which is an elastic net that essentially goes in and asks which marks are the best predictors together of a potential outcome. And the outcome they're looking for here is chronological age or potentially mortality risk. And when they do this, they have access to potentially to individuals from birth all the way out to over 100 years old, so centenarians, a sample size exceeding, in some regards, some of these newer clocks are over 10,000 individuals. And then they look at what the predictive error rate may be in the clock in a test data set. And what they've found across a myriad of different clocks is that the performance is very, very high. They can tell you from a DNA sample how old somebody is just based on these methylation states. And this is really what the building of an epigenetic clock has entailed. And really, a lot of groups have iterated on this slightly, but fundamentally, it's really a very accurate predictor of an outcome of biological age or chronological age utilizing these epigenetic DNA methylation marks. And when we talk about this in the field, it's really referred to in terms of three different terms. Chronological age, which is how many years you've been alive. Biological age, how well your body has handled those years at the cellular level. And then the pace of aging is how fast you're aging right now, like a speedometer of aging. In terms of the different versions, so you'll start to see in the popular press and also in the literature, as I had mentioned, there's different epigenetic clocks that have been developed. And really all the difference has boiled down to is having access to better metadata that they're able to utilize in the predictions. So the first generation clocks were purely using the prediction of how old someone was chronologically, as you can see here. The second or next generation clocks and third generation clocks that they've been referred to are using data where they either have a longitudinal study design, so you can look at over time how quick someone is aging in terms of a rate of aging over time if you have access to that data. So these have been referred to as the new native pace aging markers, or mortality risk, as I had said in that Grimm age clock I had highlighted, that essentially had mortality risk prediction. So you can look at survival. If an individual is aging a lot quicker, their estimated mortality age would be a lot higher in the sense that their time for where they'll reach the end of life is nearing in terms of those potential clocks. But in essence, all of the clocks are essentially looking at the utilization of DNA methylation marks to predict an outcome of interest related to the aging process. In terms of the HIV field, at the same time that these clocks were developed, a large proportion of the publications had relied on data from individuals living with HIV, and in essence, harnessing the biospecimens that had been collected in the HIV field. And Steve Horvath, who had developed that initial clock in 2013, had worked with a group at UCLA and published work demonstrated essentially in a cohort they had looked at in people living with HIV, this concept that there was accelerated epigenetic aging. So the estimate of age based on these epigenetic clocks was greater than the chronological age in a subset of individuals that they had looked at in a cohort they had studied initially, applying kind of these algorithms, suggesting potentially that there was increased biological aging that aligned with what had been observed in a lot of the comorbidity data that had been done epidemiologically. In terms of the current use of these clocks, today I'm not going to spend time going over all the work that's been done in the field in regards to applying these epigenetic clocks across different cohorts and contexts, but there's been a real expansion of the use of epigenetic clocks in people with HIV, and a lot of groups are harnessing these algorithms to essentially see how these potentially may have application in certain disease settings or indications of people living with HIV. In terms of what's most well known currently, I think the two most interesting kind of things that have arrived from this work would be expected is antiretroviral therapy seems to restore or revert kind of this age acceleration to an effect. However, there still seems to be a lingering kind of signal of accelerated aging despite antiretroviral therapy. And some beautiful work that had been done in some cohorts where they're able to initiate antiretroviral therapy at the most immediate time during acute infection has shown that potentially these aging processes in the immune system are set up very early. So despite being able to initiate ART at the earliest possible time point, there does seem to be kind of this lingering signal that's still there. So, in these studies are kind of now looking at potentially over 20 years, what this may look like in terms of following people longitudinally over time. But I think in terms of the expansion of these clocks, in terms of research studies, has really, I think boomed in the last couple of years, and we'll start to see even more work applying these clocks across different cohorts. I do want to highlight some cautions in the application of these epigenetic clocks and potential generational difference in the epigenome. Here, I'm just giving kind of three cases of three different groups of individuals living with HIV in terms of how these clocks are applied, and we start to think of the aging process. I'm putting this here in the context of people living with HIV, but this also applies in terms of the larger neuroscience field, in terms of generational differences, in terms of the aging process and what people have been exposed to, what their life experience has been, and in terms of potentially, their history, in terms of medical history, in terms of how treatment and other impacts would alter essentially these molecular light switches I told you about and potentially alter the aging trajectories and how we interpret data. Here I'm just giving three cases, which would be, I think very, very relevant to when we start to think about applying these epigenetic clocks. And it was nice to hear the question earlier about how we would think about pediatric um, acquisition of HIV and how that may differ quite substantially of living 20 years from pediatric acquisition versus someone, potentially in their 20s that acquired HIV living 20 years, or someone at an older age over 50 that potentially, has been living with HIV for 20 years. That, what they were exposed to before that or what they're exposed to after would definitely alter the epigenome and our interpretation of potentially these epigenetic clocks. So just to provide some context here, that the caution in terms of when we start to talk about the application of these clocks, it really depends on the cohort and individuals where we're studying them. And in terms of the popular press, I think the field at large, there's really been a boom in terms of potentially the utility of applying these clocks to measure biological age, and the idea being that this may be a potential complement to other aging measures to tell someone potentially how well they may be doing in terms of biological aging over time, or potentially as a way to monitor if there's some sort of substantial change in aging that may have occurred in terms of these potential biomarkers currently has, I think really been in the current conversation in terms of aging and how we would best measure it. In terms of some criteria, the field is really working hard now to rigorously define what a good biomarker of aging must do. In the next summary slides, I'll briefly just tell you about some of the criteria that we're starting to think about as mandatory for being able to potentially use some of these biomarkers of aging outside of the research realm and potentially think of how they may have applications, in terms of clinical potential insights or potential utilization besides research at this point. So some of the criteria I think that are quite important are these biomarkers of aging, or if we think about an epigenetic clock, it would need to correlate with chronological age, which, the epigenetic clocks have done. It would need to predict potentially all-cause mortality or survival, predict frailty and or health span, and then predict the incidence of multiple age-related diseases. These are some criteria essentially that have been kind of demonstrated for some of the epigenetic clocks, but I think would be very important in terms of an aging biomarker that would receive a widespread use. In terms of other aspects that have been discussed as well in terms of the neuroscience field, it would need to reflect causal aspects of aging. So essentially what we're aging would need to actually causally potentially be linked to the aging process itself. So when I had mentioned immune senescence, a marker that is capturing that feature would be very important to think about. It would need to respond to factors that accelerate aging. So when we have potential cohorts that are driving the aging process, this marker would need to be responsive to that. It would need to respond to putative geroprotectors. So these are features of potential therapeutics that would protect or slow down the aging process. So essentially things that have been linked to improving health span, such as exercise, potential biomarker of aging should be responsive to that. And then most importantly, it would need to be validated in diverse populations. So currently, a lot of the epigenetic clocks and markers, as you could imagine, have been developed in the US and really centric in terms of the cohorts that have had access to blood specimens and the genomic techniques to look at these epigenetic modifications. As we start to think about rolling out potentially some of these in other cohorts or outside of the research realm, there needs to be a large data generation that adequately captures, the globe to really pull the data together to get a very accurate kind of estimation of aging that would not be very regional specific. So potentially where we would need to go for biological age as a potential clinical marker, it'd be nice to have prognostic risk prediction, diagnostic ability as well. How old is the patient biologically? Predictive. So, if an intervention is run and potentially for selection to target aging, will the individuals that are at a higher aging rate or age acceleration be those enrolled in a clinical trial? I think would be an important application for a potential clinical utility for an epigenetic clock or biomarker of aging. Other aspects such as pharmacodynamics in terms of responding to the intervention and potential or drugs. The one I think that's received the most attention, which I won't talk about today, has been GLP-1 therapy and how that could play a role in terms of responsiveness. And then recurrence. So essentially, if someone does have a response, or in regards to, I think cancer or other things, would the aging process essentially be a good marker to be able to see that or predict that over time? So these are potentially some of the things that are being developed as we think of rolling out potentially the further development of these research epigenetic clocks into potentially some clinical utility that might complement existing clinical tools. So just to show briefly evidence map of what's been known from epigenetic clocks across the different generations. Here I'm showing you where there's been evidence in the literature that's either been strong with the green notation or in yellow moderate evidence across the three generations of clocks. As you can see here, the second generation mortality clock, so that Grim age clock I had shown, there's been a lot of evidence across clinical domains that it has utility with being able to have evidence to be able to predict things such as frailty, nervous system, cognition and dementia outcomes, depression, psychosocial adversity, socioeconomic stress, cardiovascular disease, diabetes, kidney disease, clonal poiesis, all-cause mortality, as I had mentioned. But as you can see here, there's certain gaps in regards to be able to predict other potential outcomes in terms of these epigenetic clocks. In terms of muscle health, it's been really the third generation clocks that have captured that. But the first and second generation, there's either weak or absent evidence. But just to kind of show you that groups are working quite hard now to map potentially how these aging biomarkers may show kind of associations or ability to be able to map to certain clinical domains of interest. And then just to end today, we are looking at Reprieve in terms of its ability to have quite dramatically altered clinical treatment of people living with HIV by statin use to prevent cardiovascular outcomes. And we are currently now looking at potentially applying some of these epigenetic clocks in collaboration with Steve Greenspoon and the Reprieve team. This research is ongoing, but I just wanted to highlight it in terms of what we're potentially looking at. Potentially do statins alter these aging processes and have any associations with reducing cardiovascular events or some of the clinical measures in Reprieve that were measured. So these are things that we're currently working towards, and hopefully, I think giving some context for the HIV field and community in terms of what potentially statins may be doing for the aging process and contextualizing that in regard to epigenetic clocks. We have observed in a small pilot study, which has been published, the references at the bottom, that despite the participants that had enrolled in Reprieve, in terms of looking at 100 individuals, the majority of all those individuals that were enrolled, despite being quote unquote, no high cardiovascular disease risk and quite, I think young in regards to what we think about in terms of the aging process overall. These individuals did see an increase in terms of this mortality risk prediction at entry being enrolled in Reprieve. So these are things that we're currently now looking at and analyzing further. But I just wanted to highlight this at the end to kind of give the group some insight and things to look forward to in terms of application of these epigenetic clocks and these biomarkers of aging in regards to some well-known, I think, HIV cohorts that have really altered clinical practice and treatment of people living with HIV. Here's the paper if groups are interested in looking at the pilot study. And we hope to basically run this on the whole sub-study of Reprieve in the next year and see essentially what this looks like in Reprieve.
Thanks so much.
So just to summarize today, I told you about the field of geroscience and hallmarks, a little bit about epigenetic clocks in terms of these first generation and second generation clocks. Hopefully the group now is aware of how these clocks have been developed and how groups are thinking about improving them. And then in terms of the use of application of these epigenetic clocks in people with HIV, they essentially initially were applied very early on, and now they're really being, I think, widely applied, and we're still waiting on essentially whether these clocks can be applied or which ones may be relevant into clinical potential care or even utilization in clinical trials. And the field is working towards addressing some of the gaps that remain in validation or application of these epigenetic clocks. But thank you for the time and happy to take questions.
Thank you so much, Dr. Corley, for a great overview of the science of aging, and that prepares us to discuss what comes next in terms of interventions. But just to get us started, so do you think these epigenetic clocks are ready for prime time? Are you seeing them in routine clinical use anytime soon? How can they impact our surveillance screenings, and will they be on towards use for denying insurance and other things? A lot of
Yes. Yeah, so
questions.
currently, I think in terms of widespread spread use, the jury is still out in terms of whether that's going to occur. I am aware currently of utilizing potentially these markers as a surrogate measure for addressing some of the longevity studies that have been currently proposed. And this has really been, I think, spearheaded by the NIH and their ARPA-H initiative. So this is like a rapid deployment to try to gain FDA approval. Um, that potential effort, I think, is really going to rely on what they see in regards to whether it's able to proxy some of the World Health Organization's concepts of intrinsic capacity. So, whether we actually have to measure the clinical outcomes, such as frailty and other things, versus take a blood sample and be able to biomarker that, I think is
Mhm.
going to be what they're working towards. But currently, they're really, I think, still in the research realm in terms of not being able to be utilized as a replacement completely, but more a complement at this point. I still think we need more data and further development in terms of being able to widely apply them potentially clinically.
Wonderful. There's a number of questions on aging. I think that all of us are fascinated, or maybe should I say scared of aging. One question is whether all hallmarks of aging are altered in HIV. And then what is the gold standard for age prediction in all the clocks and other things that you presented? Is that one that you would think it should be considered a gold standard? I bet not, but I'll let you answer.
Yeah, so I think in terms of the hallmarks of aging and people living with HIV, I think there's been beautiful work, for those that have, I think written reviews in regards to HIV and aging in terms of showing
Mhm.
evidence of certain instances where certain hallmarks have been observed to be perturbed in people with HIV. It
Mhm.
is intriguing when you start to think about at an individual level, certain individuals do show resiliency as well. So today I didn't go too deep into that, but at large, in the group of people with HIV, there is the ability essentially to find evidence in the literature of a hallmark that's been perturbed in a particular cohort. But in a certain individual, whether all the hallmarks are altered, that is something that, has not been comprehensively looked at. It's something that I think people are thinking of as we build kind of these omics data sets to really have a deep understanding. But I do feel like in certain individuals, it looks like things are tracking together. So if you have increased epigenetic age, those individuals, if we measure telomere length, they have shorter telomeres. The shorter telomeres, they have immune senescence. So it seems
Mhm.
like the mechanisms of aging are tracking together. But in terms of the group at large, we do find individuals as well that are aging very well. Even better potentially than the general population. Seeing
Yeah.
those protective factors, I think, are also important for us to study in the future. And in regards to the second question, I just returned back from a Hallmarks of Aging workshop in Boston that was held. And that workshop really highlighted potentially that some of these mortality risk built clocks are the ones that have been performing the best across cohorts for being able to predict. And the slide I showed you where it referred to these second generation clocks, and you saw a lot of evidence built there across different clinical potential outcomes. It seems like there is a link between comorbidity and mortality. And if you use mortality in your actual machine learning kind of building prediction, those have been the ones that have been kind of the gold standard to benchmark against. But there is a real feeling that if more data can be included, that potentially we can go beyond what's already been shown. So that's really where the field has been spending a lot of time in terms of adding additional data sets, such as, imaging potentially and other
Yeah.
markers
Yeah.
clinically to, I use that in the modeling as well.
So one thing is to look at the clock and say, oh my goodness, it's advanced. Now, can anything be done? Can you do something? Any intervention that if you have a real marker of epigenetic age advancement that shows advanced aging, and you have the intervention, and then you measure again and you see change. So can such a thing be envisioned?
Yeah, so that's currently what the neuroscience field has been spending its most time on in terms of identifying the interventions that are moving the needle.
Mhm.
Currently, the best evidence has been what's been discussed the most from the aging field. It's diet and exercise is the benchmark. So in an individual that does show increased aging, if they don't have the most optimized
Mhm.
diet and exercise,
Mhm.
that has a dramatic effect on a lot of these clocks. In
Mhm.
regards to the therapeutics, there has been a lot of attention received, for metformin and other metabolic modulators such as GLP-1. I would say the jury is still out in terms of responders, non-responders, and then in regards to reprieve, statins as well is kind of, something we're very interested in getting, a little bit better evidence to show that potentially it is having an impact in individuals that have, an accelerated aging phenotype. Can they
Mhm.
decrease potentially? But yeah, that's how these marks are currently being utilized in terms of proxies to figure out what's moving the needle for those individuals that aren't, at an optimized kind of level. But in terms of what's optimal, I still think we still need standards in terms of for certain populations where, there is kind of a general population where people sit, generally speaking, because, your biology might be that you might have genetic aging genes that, no matter what you do, your set point is different than someone else's
Exactly.
set point. So
Yeah.
these are things I think the field is still working towards developing.
So diet and exercise, that's kind of boring. Okay, and then let's put statins in drinking water and GLP-1 receptor agonists as well
Yeah,
as
I'm not a
metformin, and
I,
everyone
I
will
I
be
go with
fine.
the firm clinical evidence. There is a group that is you know, the biohacking slash longevity field group, which is yeah, they're trying things, but the evidence is clearly not there at this point, I think to make those recommendations. So,
You so much. This is a fantastic foundation in the science of aging that sets the stage for the rest of the course. And I will pass it on to my co-moderator, Dr. Erlandson.
thank you so much.